Imagine a microscopic army that patrols our cells, targeting invading bacteria with precision. Today we uncover how these tiny peptides detect, attack, and dismantle microbial foes. Antimicrobial peptides are short proteins produced by many organisms, including humans. They serve as first responders in the innate immune system, acting before adaptive defenses arise. These molecules typically contain 12 to 50 amino acids and carry a net positive charge. The positive charge allows them to bind readily to negatively charged microbial membranes. Upon binding, they insert into the lipid bilayer, forming pores that compromise membrane integrity. This pore formation leads to leakage of ions and small molecules, ultimately killing the pathogen. Some peptides also disrupt intracellular processes after crossing the cell wall or membrane. Human cells release these peptides into surrounding fluids, such as saliva, skin surface, and mucus layers. The rapid action of antimicrobial peptides helps prevent infections before they establish. Their broad-spectrum activity covers bacteria, fungi, and some viruses. Because they target fundamental structural features, resistance development is relatively low. Understanding their mechanism informs the design of new therapeutic agents. Pause 0.8. Antimicrobial peptides are short proteins that vary widely in amino acid composition. The most common classification groups them into cationic, amphipathic, and cysteine-rich families. Cationic peptides carry a net positive charge, enabling electrostatic attraction to negatively charged bacterial membranes. Amphipathic peptides form helices, with one hydrophobic face and one polar face, allowing membrane insertion. Defensins are cysteine-rich peptides that use disulfide bonds to lock a compact scaffold. The rigid structure of defensins positions key residues for pore formation or ion channel disruption. Allele37, an amphipathic peptide from humans, adopts an alpha helix that can wrap around lipid bilayers. Meganin, a cationic peptide from frog skin, forms beta strands that insert into membranes to create transient pores. The 3D ribbon models show how the spatial arrangement of charges and hydrophobic patches dictates function. When peptides align on a membrane surface, they can oligomerize into toroidal or barrella-shaped structures. These structural motifs determine whether a peptide disrupts membranes, interferes with intracellular targets, or both. Thus, the chemical architecture of each peptide class directly informs its antimicrobial mechanism. Pause 0.8. Antimicrobial peptides carry a net positive charge that guides them toward bacterial surfaces. Bacterial membranes are rich in negatively charged lipids such as phosphatidylglycerol and lipopolysaccharide. The electrostatic attraction between the peptidia-Eurostrade-S positive charges and membrane lipids initiates contact. This initial binding aligns the peptide with the lipid bilayer, positioning it for insertion. The Peptidia-Eurostrade-S amphipathic structure allows its hydrophobic face to embed into the membrane core. As more peptide accumulate, they form transient pores that disrupt ionic balance. The resulting loss of membrane integrity leads to bacterial cell death by lysis, or metabolic collapse. Because the mechanism relies on charge interactions, it is effective against a broad range of microbes. This electrostatic targeting also limits damage to host cells that lack similarly charged membranes. Thus, the positive charge of antimicrobial peptides is a key determinant in their selective membrane attack. Pause 0.8. Antimicrobial peptides target bacterial membranes by inserting themselves into the lipid bilayer. In the barrel stave model, multiple peptides align to create a cylindrical channel that spans the membrane. Each peptide contributes hydrophobic faces to line the pore interior, while charged residues anchor it to the surface. The toroidal pore variant shares this alignment, but also bends the lipid so the pore is lined by both peptides and lipid head groups. Both pore types allow ions and small molecules to leak, collapsing the proton motive force of the cell. The carpet model differs in that peptides coat the membrane surface like a blanket without forming discrete pores. As peptide density increases, the bilayer becomes destabilized and fragments into micelles. This micellization sequesters lipids away from the bacterial envelope. Electrostatic attraction between cationic peptides and negatively charged lipid head groups initiates both mechanisms. Hydrophobic interactions then drive peptide insertion and subsequent structural rearrangements of the bilayer. The dual strategies ensure that a single peptide can adapt to diverse membrane compositions. Understanding these models guides the design of synthetic peptides with enhanced antibacterial potency. Pause 0.8. Antimicrobial peptides can traverse the outer membrane of bacteria by exploiting electrostatic interactions. Once inside the cytoplasm, they encounter a variety of intracellular targets. Some peptides bind directly to ribosomal RNA, blocking the assembly of functional ribosomes. Others insert into the DNA helix, preventing the unwinding necessary for replication. A third class interferes with RNA polymerase activity, halting transcription of essential genes. These interactions are highly specific and depend on the peptidia Eurostrades amphipathic structure. The result is a rapid shutdown of protein synthesis, which is lethal to the cell. Because the peptides act inside the cell, they bypass many extracellular resistance mechanisms. Their small size and positive charge allow them to navigate crowded cytoplasmic environments efficiently. Thus, antimicrobial peptides serve as versatile intracellular weapons that target core bacterial processes. Pause 0.8. Antimicrobial peptides are produced by host cells in response to pathogen-associated molecular patterns detected through pattern recognition receptors. This detection triggers signaling cascades that activate transcription factors such as NF-kappa B and AP1, which bind promoter regions of AMP genes. The resulting inducible expression allows the peptide levels to rise rapidly during infection, while remaining low under steady-state conditions. Many bacteria have evolved resistance strategies, including secretion of proteases that cleave AMPs or modification of membrane lipids to reduce peptide binding affinity. Efflux pumps can also expel peptides from the periplasmic space, diminishing their intracellular concentration and effectiveness. In laboratory studies, resistant strains often display mutations in genes encoding these enzymes or transporters, illustrating the selective pressure exerted by AMPs. Understanding these mechanisms informs the design of synthetic peptides that evade degradation or target conserved membrane components. One promising application is the incorporation of AMP-derived motifs into polymeric coatings applied to medical devices and industrial surfaces. Such coatings can release low concentrations of peptide over time, preventing initial bacterial adhesion and subsequent biofilm maturation. Comparative charts show that surfaces treated with these peptides exhibit markedly fewer viable colonies than untreated controls, underscoring their potential as a non-antibiotic antimicrobial strategy. Pause 0.8. AMPs are short, positively charged peptides that recognize bacterial membranes through electrostatic interactions. They disrupt integrity by forming pores or coating the membrane, and some penetrate cells to inhibit essential processes. Host regulation governs their production, while bacteria can develop resistance. Research continues to exploit AMPs for novel antimicrobial technologies.